The thing I enjoy most about my role and my job is if I've been supervising a student on something very technical, like a practical element of some research that they're doing, and then so you show them like kind of the ropes of how to do something, and then they go away and do it, and then it works, and then they come back to you with like some really cool, exciting data that they're really passionate about, and you can see that you've had an impact on someone, and it's kind of perpetuating that like positive feeling in science. Science can be hard sometimes because things don't always work, and it's not just in those moments when things work for yourself, but I find when it's working for someone else and you kind of act as a role model or mentor for them, that's really rewarding. One thing you certainly get to do, but this is possibly not so plant science specific, is you get to travel a lot in science, and I think it's remarkable how many different people you meet from different backgrounds, who've had completely different lives to you, and getting that exposure to other people and other perspectives, I think, is not just like good for you as a scientist, but just as a human being and as a person. Like, I think it helps you feel connected with the rest of the world. So I know that's not like a plant specific example, but it's certainly like my perception of working as a scientist. So my plant science career, my interest in plant science began, I'd suppose, at the start of my PhD. So that was really when I became immersed in an environment of lots of different plant science, specifically looking at like plant microbe interactions. And it was, during that time that it kind of really stuck home to me how like cool plant science was. I actually wasn't specifically motivated to apply for a plant science PhD. I was more interested in like kind of the cellular systems and biology in general, and how proteins interact and how that can facilitate changes at the cellular level. And it kind of just so happened that was in a plant system, but then ever since then I've been totally hooked. So my current area of research is looking at how the structure of DNA, specifically the structure of chromatin, is able to facilitate long-term memory of pathogen resistance. So we know that the accessibility of genes through the compaction of this structure called chromatin can either increase or decrease gene expression. And what's really cool about that is that those the way that that is structured is regulated can last for a long period of time, and it can change in response to specific signals. And the research that I'm conducting now is looking at how we can use those signals to imprint a memory of pathogen response in plants which have never experienced disease, so that they are more resistant to disease. And that's really important because so much of our crops and our food production is lost to diseases and pests every year. So I think one of the hottest topics in research now, which is really still in its infancy a bit, I would say, is how we can use gene editing techniques like with CRISPR-Cas9 systems to actually change the structure of DNA through histone modifications or DNA methylation. And that has really come on, like CRISPR technology has come on so far, so quickly. And, it was first used as a tool to just introduce mutations, and now we're able to use it to actually change the tertiary structure of DNA through post-translation modifications. And that's like amazing, because then we can really have systems where we can control genes which are on and off in response to like external stimuli.